Okay, so first of all, I want to thank you, the organizer, for giving me this great opportunity to present my results. So, as mentioned by you, I will talk about MIR-663a, that is modulated by BEF1, a gene encoded by Epstein-Barr virus, also known as EBV. So EBV is a gamma herpes virus and is belonging to the herpes family. It is a double strand of DNA and is mainly transmitted by saliva. However, other biological fluids may be involved in this transmission. The virus impacts more than 19% worldwide, and in most of the cases, this virus causes an asymptomatic infection. However, in a percentage of cases, the virus is associated with mononucleosis, with immunosuppression, and epithelial and malignancy with epithelial or lymphoid origin, as Hodgkin lymphoma, nasopharyngeal carcinoma, and burkitt lymphoma. The virus establishes its latency in the host cell and produce more than 80 viral proteins and more than 40 viral miRNAs. miRNA is a small non-coding RNA, in generally is 20-22 nucleotides, that is able to negatively affect the expression of host gene expression by binding the mRNA target and induce a translation repression or degradation. As you can see from this picture, miRNA may have many cellular targets and can be also target of other miRNAs. In general, one of the most well-studied is very important because it helps to with the differential diagnosis. For example, is very high expressed in diffuse large B-cell lymphoma and help us to distinguish the two type of forms of diffuse large B-cell lymphoma, ABC and germinal center, but it also helps to distinguish diffuse large B-cell lymphoma from other lymphomas like follicular lymphomas. So the miRNAs are very important for the diagnosis and are easy to detect as they are present in blood, urine, and other biological fluids. And these miRNAs are also important for the treatment because if we target just one miRNA, we can target many pathways. So regarding the viral proteins, the virus is able to encode non-coding RNA, long non-coding RNA in this case, like EBRs, but also long coding RNAs like EBNA1, EBNA2, EBNA3, LP1, LP2, and all other genes that are reported in this picture. Based on the expression of these viral proteins, the virus establishes its latency that can be called latency type 1, latency type 2, and latency type 3. These latency are characterized by the presence of EBRs and miRNAs that are always expressed, while the latency type 1 is also characterized by the presence of EBNA1 only. And this type of latency, it is we can observe this type of latency in burkitt lymphoma, for example. Latency type 2 is observed in gastric cancer, in nasopharyngeal carcinoma, in Hodgkin lymphoma, and is characterized by the presence of LP1 or LP2 other than EBNA1. The latency type 3 we can observe in post-transplant patients, so when the immune system is not working very well, and the virus can express an highest number of viral protein, like EBNA1, EBNA2, EBNA3, as reported here. And I will talk about the Burkitt lymphoma, as mentioned by the title of my presentation. Burkitt lymphoma is aggressive B-cell lymphomas, and is divided into three forms: endemic, sporadic, and immunodeficiency associated. All these variants are characterized by a translocation involving the protocogen MYC and the immunoglobulin, leading to an hyperproliferation. At histological level is characterized by a high number of macrophages that give to the tissue a stereoscopic appearance. And regarding the clinical level, the disease is present with a local oral localization, but also other organs are involved. And the association with EBV, it depends on the forms of Burkitt lymphoma. For example, in the endemic one, we can have a strongly association with EBV that is present in nearly all cases of endemic Burkitt lymphoma. While in the sporadic form, we have a low EBV association, and in the immunodeficiency associated PL, we can find that around 40% of cases are EBV positive. So as mentioned before, in Burkitt lymphoma, EBV is just established a latency type 1, characterized by the presence of EBNA1. However, we for the first time showed that the Burkitt lymphoma cases may also express a higher number of viral proteins. That includes, for example, MP2, BMR1, BZLF1, BHRF1, other than BIF1. And as indicated in this picture, BIF1 is very high expressed and is expressed in almost all cases of Burkitt lymphoma. So this gene escaped to the attention of the scientific community, but is very important because it is an active viral G protein coupled receptor. We know this gene is largely unknown, so just few papers are available. And what we know is that it is reported as an oncogene because it's transforming power in nude mice, and it has a role in immune response as it will decrease the MSC type 1 levels and lead to the virus an immune evasion from the T-cell recognition. So based on that, and based on the fact that BIF1 is a viral G protein coupled receptor, and consider the fact that the viral G protein coupled receptor are target of therapy for the other viruses, we decide to investigate the expression of these genes in a larger cohort of Burkitt lymphoma and also in B-cell lymphomas. And we also try to define its impact on transcription targets encoding cellular memories in B cells. So regarding the first point, we detect the presence of EBV encoded BIF1 in B cells, in this case in Burkitt cell line, sorry, in this case we used Hakata, and we used by using an RNA-scope assay. This RNA-scope, this technique is a novel approach that led us to identify the presence of mRNA in in the cells, and it is a in situ hybridization. The characteristic staining of RNA-scope is a red puntate dots or a brown puntate dots, as in these cases. And as you can see from the picture, we detect a strong expression of BIF1 in almost all the cells of Burkitt's lymphoma cell lines. And this technique, you can see from this, but this technique also let us to localize the signals that were mainly near the nucleus. So we collected 28 cases of Burkitt's lymphoma, and we investigated these cases by using a fluid PCR. As you can see in this picture, we detect and we investigated the presence of all viral proteins of EBV, including BIF1, and as indicated by the red arrow, all cases except two expressed BIF1, and BIF1 was also very expressed, strongly expressed in most of these cases. So we confirmed this data also by RNA-scope, showing the presence of a BIF1 mRNA in almost of all cells. So we define the impact of this gene in B cells, because are the presumed progenitor of Burkitt lymphoma. And to do that, we isolated B cells from fresh tonsils. We transfected these cells by with BIF1 plasmids, and then we collect these cells to perform RNA-seq. In parallel, we reanalyzed cases published by our colleague Francesco Abate, in which we detect a non-canonical latency of EBV. And in the end, we compared the list of genes detecting the dysregulated genes by BIF1. So regarding the regarding the genes that we detect from RNA-seq data, we found that BIF1, when it's expressed in B cells, is able to dysregulate many cellular genes, more than 6,000. And we looked at the function of these genes and the related pathways. And as you can see from this picture, these genes, target of BIF1, were involved in important cellular pathways like targets, like metabolism, like cell proliferation. And we did the same for the genes that were significantly associated with a strong expression of BIF1 in the paper Francesco Abate. And again, we detect important cellular pathways that were dysregulated. Some of those were overlapping. So we compared the list of genes described to be dysregulated by BIF1 in B cells and the genes that were dysregulated in primary tumors by BIF1 as well. And as you can see, more than 500 genes were in common between these two sets of data. Among these genes, we focus our attention on MIR-663a host gene. And this gene is located on chromosome 20 and encodes for a miRNA called MIR-663a. This miRNA is well described in other disease and is involved in, is described as an oncomir because it's involved in the pathogenesis of disease like colon cancer, nasopharyngeal carcinoma, like a renal cell carcinoma. But its role in B-cell lymphomas in general and in Burkitt's lymphoma especially, it has been not studied. So we decide to confirm first of all the expression of this gene and is related to MIR. And as you can see from this graph, we detect a strong expression of MIR-663a gene in the cells that were transfected with BIF1, and we also detect a higher expression of this MIR after BIF1 transfection. We also observed that this gene was overexpressed in all or almost all the cases of Burkitt's lymphoma. Then we did the same for MIR-663a. We checked its expression in B cells and in primary tumors. In B cells, we observed that this MIR was higher regulated after BIF1 transfection in B cells, as you can see from here, and the difference was significant. And we also observed that this MIR was highly expressed in the same set of Burkitt's lymphoma. So we define the rule, we better define the rule of MIR-663a in primary tumors. And to do that, we focus our attention on predicted targets and on validated targets. So regarding the predicted targets, we applied a bioinformatic approach. So we used 4 platforms to perform this analysis that are called Target Scan Human, MIR-based, MIR-RAND, and MIR-Gene. And we selected the genes that were in common to these 4 platforms. So we selected more than 800 targets, and we looked at their role and the pathways in which these genes were involved. So I have indicated in red some important process that regarding the viral process, so the antiviral defense of the host cell, and also the antigen processing and presentation of peptide via MHC type 1 that is already described in the literature because BIF1 may have a role in its decreasing. So we did the same for the validated targets, and these targets were validated in both in vitro and in primary tumors, as you can see from this picture that we took from a Diana tool. And indicated in red are the processes that are in common to the predicted target here, but also pathways very important in the pathogenesis of Burkitt lymphoma because it included mature B cells differentiation. So what we have done is an additional comparison between the genes that were described to be predicted by MIR-663a, the target genes that were validated and described to be target of MIR-663a, and the genes that were dysregulated and in particular downregulated by BIF1 in B cells. So as you can see from a Venn diagram, more than 140 genes were in common to these sets of data. So BIF1 and MIR-663a share some of gene targets, and we selected these genes on the base that based on their role, because they are involved in immune response in B cell differentiation, that is very important in the case of Burkitt lymphoma, and also regarding the MHC type 1 antigen processing, because it also have a role. It also has a role in the pathogenesis, in the lymphomagenesis. So we further investigated these genes by digital PCR and confirmed their low expression. And we also we are now performing the immunohistochemistry for these genes. So in the conclusion, we can say that BIF1 is expressed in Burkitt's lymphoma, and BIF1 induces MIR-663a host gene hyper expression of MIR-663a host gene expression, and which in turn will lead to give an hyper expression of MIR-663a. So in the conclusion, we can say that we define a new rule for BIF1 in the pathogenesis of Burkitt's lymphoma, because BIF1 will induce this gene and this related MIR, which may play a substantial role in the etiology of Burkitt's lymphoma by regulating networks involved in B cell proliferation and differentiation. So I want to thank you, my supervisor, Professor Paul Murray from the University of Limerick, but also my colleague from University of Siena and the European Committee that granted this project with a Marie Curie fellowship. So thank you for the attention.